Hello, students. Hi, how are you? Okay, I'm very happy to see you today. Okay, today I have a very interesting lesson for you. Today I want to finish chapter 6, which is the sexual reproduction of flowering plants. Okay, I have a lot of interesting facts to share with you today in today's lesson. Okay, first of all, can I have a feedback from you whether you're able to hear me, whoever is following my class now? Okay, and let's check on your comments. So if you are able to hear, okay, Sao Yi, right? Hi. Okay, nice to see you here. Okay, can you tell me whether you can hear me? Okay, if there's any problem with the audio, Sao Yi. Okay, just say yes, can hear. All right, fine, that's good. Okay, Yun Chi, hi. Yeah, girls. Nice to see you. Okay, so today I'll get straight to the lesson because I have a lot of interesting things to share with you. Okay, now, but before that, I would like to answer. Yeah, okay, thanks, Saoyi. I'd like to answer Saoyi's questions last week. Okay, remember that she asked some two interesting questions, which I find they're very interesting. Thanks for asking them. Okay, by asking questions, actually, you get to know more things. All right, so I would like to share with you my answers and what I have found. All right, let me open. Oh yeah, it's open. Okay. Right. So here, all right. You can see the screen, I hope. Okay, I have shown this pollination and fertilization. So this is today's lesson. But before that, I would like to go and tackle the question that was given last week. All right, last week, so you asked whether potatoes have seeds. Okay. Now the answer, yes, potatoes do have seeds. Okay. So now potato seeds, there are actually two different definitions. When you talk about potato seeds, okay, either you're referring to the real seed. Okay, now why do potatoes have seed? It's because they actually have flowers. So I wanted to show you the flower of potato. We don't see the flower, we always see the potato, correct? We just buy from the store and that's it. So we are used to the potato tuber. Okay, so let me just check whether I am looking. Yeah, all right, you're looking at this. Okay, so this is the potato tuber, which we dig up from the ground, and then we eat that because it's full of starch, full of carbohydrates. That's where the storage of the food is. But we rarely see the plant itself. Okay, now this is the stem. All right, you can see, I think hope you can see my mouse, right? So here is the stem, the green part of it, and this is the potato flower on your left. So there could be purple, could be white, okay, or shades of purple or white. So it's very rare to have red potato flower. Then this is the fruit. You do have a potato fruit. So potato fruit looks very much like a tomato, but the tomato is very different. Do not eat the potato fruit because it's known to have toxin. So you're not supposed to eat the fruit. Okay, potato fruit. Now inside the potato fruit, you have the potato seed. So this is what it means by the potato seed. Okay, now you can actually, theoretically, you can plant a potato using the seed. But of course, you know, it will take some time. Now, farmers don't do that. Farmers do not use the seed to plant for two very basic reasons. Okay, I'll tell that afterwards. Okay, let's go back to the potato. So potato, actually, they do have seeds, right? So they are flowers. So when you ask yourself a very simple question, whether the plant has seeds or not, ask yourself whether it's flowers. If it has flowers, it will have seeds. And you have seeds, it means it have fruits. So all these three are interconnected. So ask yourself, if you ask yourself whether tomato is a fruit or a vegetable, it's always a perennial question, whether it's a fruit or a vegetable. Ask yourself a very simple question. Does it have seeds? Does it have a fruit? Okay, let's say very easily, tomato has seeds. So if tomato has seed, it means it has to have fruit. Only fruit bears seeds. And if you have fruit, that means there would have been flowers in the beginning. Okay, so there is tomato flower, there is a tomato fruit, and there is a tomato seed. Okay, now let's go back to the potato. Now, potato, the real seed, this is the one that we find inside the green color thing. We saw that is a potato fruit. Okay, but then there's another way of saying potato seed, which farmers use to plant. This is not really the potato seed. This is the potato tuber. Now, this is the potato tuber that we eat. Okay, now this is the tuber, all right? This is the one. Wait, this is the one that we eat. We buy the potatoes. And we have the eyes there, isn't it? The eyes, we call it the tunas, or where it's going to germinate into a plant. So if you do not eat it, right, it's going to germinate, you're going to baby plant. So what they do is they cut up this pieces of potato, cut up into small pieces. At least each piece have an eye. Now that would be called a potato seed. They actually plant that, farmers plant that And from there, you will get the whole potato plant later. And that potato plant, in the roots there at the tuber, you will have many potatoes later on. Okay, so that's how they plant potato seed. Now, why they don't use potato real seeds to plant? Okay, so coming back to what you've learned in Form 4 last year, when you talk about mitosis and meiosis, when you use the seeds, when you plant from the seeds, this is actually the seed that that germinate from there, the plant that germinate from the seed here is actually not genetically the same as the parent. Okay, so that means you could have a different kind of quality. When you plant from the seed, whenever you plant from the seed, from a fruit, it will never have exactly the same qualities as the parent itself, because this is sexual reproduction. As you learned earlier, sexual reproduction means that there is variation. Okay, you have the crossing over and everything. So the result of that, you will have gamete. And when male and female gamete fertilize, you have a zygote. So that zygote will carry characteristics from the male plant and the female plant, and it's not exactly the same as the parent plant. So you see why we don't use it. Okay, so what is a true potato seed? The true potato seed is the one that you found in that fruit, that potato fruit. But farmers don't use that. Okay, now why? Because number one, it takes a longer time to grow. All right, it takes a longer time to grow, and you will not get the same quality from the with the parent plant. So you see, like, we'll have different characteristics than the parent plant. They are not clones. But when you plant from the tuber itself with the what they call the eye, you actually get a clone of the actual plant, because it's vegetative reproduction. You remember in Form 3, right? We have learned vegetative reproduction, or even in Form 4 bio, right? When you go to that part on the mitosis. So that you will get a plant which is exactly the same as the parent being the one that is cut up lah. Okay, so first of all, you get the exact one, right? And it will grow faster if you plant it from that potato tuber. Okay, so how to plant it, or if you're interested, then you can read up this one. So I'm not going to go into that because it'll take some time. You can just look at the video later on, pause it, and just read it. How do you plant potato from potato itself? You can get a whole tree because it is vegetative reproduction. Okay, so that answers your question whether potatoes have seeds. Yes, they do. They do have flowers, they have fruits, but farmers do not plant from the seeds. Okay, they plant from the tuber itself, which they cut up into smaller pieces, at least with 1 or 2 eyes, so that it guarantees that it will germinate. Okay, now second question about Partinocopy. Okay, now Partinocopy, it actually occurs naturally as a mutation in nature. So they do happen. Example is bananas. Bananas, they develop into a fruit. Actually, it's a Partinocopy. We don't spray auxin to the banana fruit, the banana tree, but they do develop into, or the ovary does develop, they do develop into a banana, we call it the banana, a part that we eat lah, all right? So actually Partinocopy comes from the word, it actually means virgin fruit. When you talk about carpi means fruit. Okay, let me see whether you have any. Okay, Rachel is here. Hi, Rachel. Okay, so interesting enough, you put a "oo" there. Many people do not know. In fact, I had to search to find out. Yeah, we rarely see the potato seed or the flower. Okay, so back to Partinocopy. So it develops fruit without fertilization. So it does not need, the advantage is it does not need to have pollination. Okay, you do not need to rely on the male gamete to come over or transfer over to the female part of the plant in order for it to develop a fruit. So it naturally actually occurs in nature as a mutation. So it's something to do with genetics. Okay, so Partinocopy is the ability to develop fruits without pollination. Okay, and it is actually controlled by an incompletely dominant gene. Okay, we will not go into that because it's not needed. So it has something to do with the genes. So we can actually, by spraying auxin, we actually increases the chance of it to happen. So by spraying auxin, not only auxin, we can spray also gibberellin or cytokinin. All these 3 growth hormones have the effect of inducing Partinocopy. So we actually make increase the chances of that Partinocopy occurring in the fruit itself, because it does occur in nature for certain species like bananas. Okay, cucumber, grape, orange. It does. It can be induced by spraying auxin. So no need to go into details about this because it's all about genes. When I was reading through it, it's all very, very, what we call very, very complex in nature. So we just need to know that these auxins have the ability to maybe speed up this natural process. So these plants actually, naturally, they do are able to, some of it are able to carry out partinocopy in nature. Okay. So it's actually a vegetative reproduction, okay, in that sense. So without fertilization and so on, okay, they also produce bigger fruits. That is the advantage of Partinocopy. Okay, now let's go on to today's lesson, okay, which is 6.3 and 6.4. Okay, if you have time, I'll go through the last little part, which is 6.5, which is the importance of seeds. Okay, now let's look at pollination and fertilization. So is pollination the same as fertilization? Okay, what do you think? Does pollination equate fertilization? Okay, what do you think? Fertilization, you're familiar with that, right? When you learn about your sexual reproduction in human last chapter, where you have the male gamete, right, which is a sperm, okay, coming into the female body through the vagina, and it goes up the fallopian tube and it meets the ovum. There you have that fertilization when the two gametes join together. You're familiar with that fertilization. A male gamete meets a female gamete. But what is pollination? You don't have the concept of pollination in humans, right? But actually, pollination is very similar to just transferring the pollen on the part, that means the male gamete to the female part. So pollination does not equate fertilization. It's not the same thing. Pollination is just transferring on it. So it's a process of transferring. Okay, transfer of the male gamete to the female organ, so that in order for the two gametes to meet up, to be able to fuse together for fertilization. So this is transferring on it. So it's very similar to humans, like what we call, very crudely, we say having sex, okay, because sexual intercourse, okay, where the male sperm is put into the female body. That's all. Pollination is simply a plant version of having sex, okay? Very simple to remember. But fertilization is a different thing. Pollination does not guarantee fertilization. You can have pollen coming over to the stigma of the female plant, which is the female organ, right? But it can go down, right? The male gamete can grow down into the tube and all that. It can find its way down into the ovum, but whether it actually fuses together is a different method. Okay, pollination just transferring, but whether the male gamete actually successfully join together with the female gamete later on is a different thing. So that is fertilization. So there are two different things actually. Okay, so do not get mixed up, do not get confused. Pollination is different from fertilization. Okay, so let's look at the text here. So pollination is the process where the pollen grains are transferred from the anther, the male organ, okay, which produces the pollen grain, to the stigma, which is the part which receives the pollen grain. So that process of transferring is called pollination. And that can be done in many ways. One of it is through wind. All right, you've learned that in Form 3 before, when the pollen grains are very light. Usually for flowers, they're not beautiful, they are not colorful, they cannot attract the insects, and then they have to rely on wind. So when the wind comes over, it blows over and it drops onto the stigma. That is pollination by wind. It can also happen through animals or insects. Okay, it can be from bats, insects, bees. Okay. Even human can also do it artificially. We can put the put the pollen grain over to the stigma. We can also do it. So that will be also pollination. So they are called pollinating agents. It can be mammals, it can be birds, it can be water also for aquatic plants, right? And also wind. Okay, so that is the difference. So let's iron that out. Okay, now let's look at what happens after pollination. So let's say this pollen, this looks very much like a hibiscus pollen grain, where it looks like very much like a mini durian. Okay, I always call it a mini durian. It looks like a durian, right? Because you have spikes there, and it is not usually pollen grains which are fertilized pollinated by what they call animals or insects. They are usually rough because they need to stick onto the stigma, and stigma has sugary solution to help to stick onto the pollen grain as well, and also to induce what we call growth of pollen tube later on. There is sugar or sucrose, okay? Sucrose solution produced by the stigma. Okay, now let's look at what happens when it comes over. Let's look at the process now, okay? Pollination has happened, all right? Now this pollen is a long way off from the gamete, which is down here. So if you look at this picture here, the pollen grain is up here, all right? And your ovule is all the way down there. So it has to find its way down. So how to find its way down? Remember when we learned about the pollen grain, the formation of pollen grain, there were two nucleus inside here. Okay, our previous lesson, we have the generative nucleus and the tube nucleus. So this whole thing is called the microspore when you have two nucleus here. Okay, the generative nucleus later on will be the one which produces the male gamete. So the genetic material is in the generative nucleus. So this is the genetic material. So we call it the male nucleus are here. The male nuclei. Now why I say nuclei? Because actually there are going to be two. Okay, so the male gamete lah. Okay, let's put it gametes. They are here. This generative nucleus is going to undergo mitosis. You're going to get two later on. This tube nucleus, on the other hand, is just predominantly for producing the tube, which is called the pollen tube. Okay, so let's look at what happens and look at the text. Number one, when it's ready for pollination, the wall of the anther from the mature pollen will dry up. Okay, it will crack, it will shrink, it will split, and it will release the pollen grains. So they are considered mature. Okay, they release it. It will look like flour. Okay, or we call it very dust particles, very very small. And some people are very sensitive to that. When they smell that, okay, they have this sensitive, we call it nose will become watery and so on. So you have sensitive nose and so on. So we call it hay fever, right? Usually happens in the spring. People say hay fever, you smell all these pollen grains and you can find you have a lot of sneeze and all that. These are people who are sensitive to pollen grains. Okay, so it comes over, now they are on the stigma. The released pollen grain are transferred to the stigma by pollinating agent. It can be insects, it can be water, it can be wind, okay, it can be anything that transfers it over. So now the pollen grains have come to the stigma. Now it will germinate. So the next stage is it will germinate. How it germinates is there will be a sticky solution called sucrose on the surface of the stigma. So that sugary solution, because sucrose, will help to induce germination. So this pollen, the tube nucleus will actually grow. Okay, it's going to form a pollen tube. The pollen tube comes from the tube nucleus. So this one makes the pollen tube. Okay, so this is the job of the tube nucleus to make the pollen tube. Okay, so the pollen tube will grow downwards and grows through the style. So this whole thing is called the style. Okay, and it's going to find its way all the way through the to the ovule, all the way down there. Okay, it's all connected. So the generative nucleus now will move behind. So after the tube nucleus move, there is going to be the generative nucleus, right? Remember, the generative nucleus is another nucleus there. Now it will move along behind it. And at the same time, once it comes down, it is going to split. It's going to divide by mitosis to form two male gametes. So you will have this one here, and they are both haploid. Okay, so there are going to be two male gametes following the tube nucleus down in the pollen tube. Okay, so it goes all the way down to the ovule. Okay, next. It goes all the way down. At the end of the pollen tube, right, it will secrete an enzyme to break, okay, to digest the tissue of the style. So it's going to digest, it's going to break open this part, okay. So the enzyme is going to, let me show you, okay, okay. So it's going to digest this, oops, sorry, disappeared totally, yeah. So this is the tube, right? And this is the tube nucleus, right? So enzyme is going to be produced, okay, and it's going to split, okay, it's going to split here, okay, so that your tube nucleus can come out. And also don't forget, your very important is these two, what? The male nucleus that follows behind. This one, the two male nucleus that follows behind the tube nucleus. Okay, now, okay, so to digest the tissue, right? Now, when it reaches the embryo site is basically the ovule. The pollen tube will penetrate the ovule through the micropyle. Remember, there was one place that you call the micropyle. There's a little lubang here, right? the integuments, there are two layers there called integuments. They do not envelop, they do not close up the whole embryo sac. There's going to be a little hole there, which is going to allow this male nucleus or male gametes to go in. Okay, so once it comes to this stage, the tube nucleus will degenerate. So it's not needed anymore. It's done its job. Okay, no need. Now it's up to these two gametes to enter the embryo sac for the process of fertilization. So fertilization hasn't happened yet. This is just going in on it. Okay, so fertilization only happens when your male gamete joins with another female gamete. Inside there, you will get a diploid. Okay, so this is one of the fertilization. Okay, now just to set things straight, what you see here is only one of the ovule. In an ovary, there are many ovules. Okay, there are many ovules, not only one. So they're only showing one, you know. So in order not to confuse you, they're only showing one. But you can see here there are 6. All right, 6 is in fact a very conservative number. There are more than 6 inside here. So one ovary can have, you know, you can see some fruits are so many seeds inside, like the papaya. So many black seeds inside there, right? So each one of them actually, all right, it was connected to a style. So each ovule has a style connected. So not only one style, no? There's not only one style. There are many styles. Each ovule is connected to one style. So take the example of the corn. You know, the bao suk, jagung. Okay, when you look at the jagung, when after the jagung is developed, you see this whole husk here, isn't it? The whole husk here. Then you can see all the rows of jagung here, all right? All these jagung, right? Now, each one of them actually was an ovule. And each one of them was connected to what? Connected to this is a style. So you see all the husk, when you peel off the husk, you see all the benang here, all these threads here that you throw away, that you don't eat, they were actually the style. Okay, so each one of these threads that you see here is actually the style, and it was connected to one of these biji here. Okay, and of course the stigma, the style here has dropped off on the top here. So now you have this whole thing, you should call the corn. Each one of them is actually an ovule. Okay, that was already fertilized. Okay, clear? All right. Okay, I'm just closing it so that I can see your comments. Okay, today I'm just using one monitor, so I have to flip and see. The other last week I was using 2 monitors, so easier for me to see. All right, so today I only have 1 monitor, so I have to flip and look at your response in case you have. Okay, so clear? Now this is fertilization. But in animals, sorry. Let's go to, we're going to do this experiment next week. Okay, those who are in my class, we're going to look at the germination of pollen grains and also formation of pollen tubes under using a sugar solution under the light microscope. So you're going to do this. So because next week you're going to have some practicals, only 1 more week remaining. So that's why I want to do the theory online class so that I can leave the class for practical. Okay, for you to do the practical. So now you see, we need to use a cover, what you call a slide, which has a cavity. So there are actually 2 types of slides. One type is a smooth one. Okay, it's actually flat. There's no curvature in the middle. And another type here is actually there's a little dent. Okay, so if you look at the surface, it's a little bit of dent there. This type of slide is usually useful for us to prepare slides if you want to see movement. For example, amoeba, paramecium, or you want to see a 3D shape of the structure. Okay, you can see more clearly. So we want to see, we want to put our sucrose solution there. We put in the anther, the pollen grains are anther, pollen grain, and we add in the sucrose solution, and then we leave it for about at least 20 minutes. Okay, so first of all, how do we do this? Take a slide, okay, place sucrose solution 10%, put about a few drops, 2 drops are not too much, into the cavity there. Then you take the stamen part of the flower and you try to take the dust, actually it's the pollen grain. Don't take the anther, anther is too large, too big. You are not able to see much, it's too thick, the light cannot go through. So you take the powder, I call it the pollen grain, try to dip it into the liquid, it's going to drop into the water, into the solution. So when you drop inside there, there will be quite a number. Don't worry because it's too small for you to see. There will be. Okay, so you drop it there, then you cover the slip, with the cover slip, cover the slide with the cover slip, and then you leave it for about 20 minutes. Okay, first you see it first. You want to see the original shape of your pollen grain without germinating. So it's going to look very basically like this. Okay, last week I've done it with my class. Okay, you're going to have all these spikes. Something like that. So I call it mini durian. Okay, mini little durian. Very beautiful. Okay, very beautiful, very geometric shape, very nice. Because if it's hibiscus, of course, different pollen grains of different species will have different shape. I'm talking about the hibiscus. It looks like this hibiscus flower. Okay, so let me check if you have any. Okay, so far nothing. All right. So you have this. You leave it there. Now, every time you use a microscope, you must start with a low power objective lens. Turn to the lowest. Okay, I'm sure your teacher will tell you that. Because if you turn to the highest or medium or high, you're not able to focus it because the range is too much. Okay, you won't be able to find a good focus. You always must start with the low power and then you turn your platform up until maximum, until you cannot go anymore. Okay, then after that, as you look down the tube, right, you turn the platform down slowly until you see an image on the in your your what we call view of vision. Okay, so now here you're going to stain. After that, you use this. You're going to stain with a coloring called acetocarmine stain solution. This is red in color. How to stain it? Okay, this is a method called this is a method of producing a stain. I mean, actually of introducing the stain. Remember, you have your slide cover already there. You have already put a slide, okay, and then it's let's say if very neatly done, right? You don't have bubbles and all that. Now, if you were to open it and you want to put your stain there, you're going to introduce bubbles into it again. So we do not open the cover when you want to add in a stain. So what you do is you put the stain at the side. Here, let's say here at the side, you drop one drop of the stain at the side, and then you use a piece of filter paper on the opposite side, and you just put it at the side here. It will draw the solution inside because of the fact, what you call that, the cohesion and adhesion, capillarity action. Capillarity, remember you learned it before, capillary action. Water will draw water, okay? It will pull the water in. So water will draw this acetocarmine solution inwards, and you'll find that this part is going to be colored red, and the excess water will be drawn out into the filter paper. So it's going to your color, or I mean your water is going to be removed, and it's going to your acetocarmine will come in. Okay, so that is the best way. You never open a slide to color it. You never do that because it's going to be like, you know, you're starting from zero again. You have to make sure you put your cover. Of course you can, right? Because you have done it already. So you don't want to open it and put it in. So this method is the best way to do it. All right, so the filter paper is to absorb the additional water and to pull the water, to pull the coloring, the stain inwards. Okay, you're going to see something like this. All right, if it is, this is not exactly the pollen. All right, I'm going to show you this is actually cells undergoing mitosis. Now, you can use it in mitosis. When you want to look at slides, they're already stained. We have prepared slides in the lab for you. So acetocarmine is a stain used to stain nucleic acid inside the cell. So as such, you are able to see the chromosomes. It is going to stain the chromosomes perfectly until you can see at what stage you can identify prophase or metaphase or anaphase or telophase just by looking at the arrangement of the chromosomes. You can see it very clearly because they're already stained. If you don't put the acetocarmine, you'll find that some of the strands here are quite transparent. You cannot see it. Okay, so usually it's used in when you want to study the stages of cell division during mitosis or meiosis. We put this thing called acetocarmine. Okay, so there's something that it's not nothing. This is not a slide for you know, the pollen grain. This is actually cells undergoing cell division here. Okay, right. So this will show you the coloring. Now, what can you use? You can use pollen grain from hibiscus. This is your bunga raya. You can also use alamanda. Okay. You can also use balsam. This is the balsam. We call it bunga tahi ayam. Okay, in Malaysia. So this is balsam plant. Now this is non-woody. Non-woody. So I'm marking my SPM paper and so on and all that. So some students, they write the wrong thing. You must use a woody plant for your, if you want to do the phloem, the bud ringing experiment. Okay, so this is a balsam plant. Okay, so now let's go back to here. After 20 minutes of immersing your pollen grain into this 10% sucrose solution, you should have pollen grain formation. So you're going to see something like this. Okay, germinating pollen tube. You can see the long string there coming out. That should be the pollen tube as seen under the microscope. Now you should see that. Okay, if you don't see that, of course, we sometimes, in experiments, we do not always get what we should get. So if you were to write in drawing, now you can see this is the pollen wall, all right, and this pollen tube has germinated, and you can see the sperm cell means this is the two male gamete lah. These are the male gametes coming out, and the tube nucleus is at the tip there. So the tube nucleus go first, followed by your two male gametes. Okay, right? Okay, so this is drawing lah, right? This is the actual picture. All right, now let's go on. Okay, I don't see any questions, so I'll just carry on. All right, so let's go on. Now, what happens at fertilization? So in plant, we call it a plant kingdom lah, right? We have a concept called double fertilization, which you've never seen before when you talk about human or animal fertilization. So in plants, you have this concept called double fertilization. What is double fertilization? That means fertilization happens 2 times. There are 2 fertilizations. Okay, now where and how? First of all, remember we said there were 2 male gametes coming down the pollen tube. Okay, so these are the 2 male gametes. Okay, look at this diagram here. So these 2 gametes have come down here. Now, afterwards, you see this pollen tube, right? It's going to penetrate into the embryo sac, and it's going to disintegrate. Your pollen tube is not needed anymore because it's done its job of making the pollen tube. Now, it's up to these 2 male gametes to do the job, which is fertilize. So why are there 2 fertilization? Because one male gamete will fertilize one thing. The other male gamete will fertilize another thing. What are the 2 things they fertilize? Remember, there were 8 cells inside your embryo sac. Okay, there were 8. Remember, there were 3 here, which are called the antipodal cells. These 3 here, they serve no function. They serve no function, just like polar bodies. They don't serve no function. These are the polar nuclei. There were 2. There are 3 here, 3 antipodal cells. Okay, these 2 here are the polar nuclei. And remember here, there are another 3 at the bottom. One is the egg cell, and another 2 are called synergic cells. They also serve no function. So this male gamete, all right, one of it will fertilize the egg cell. This one, egg cell. Okay, so I look at this bigger diagram. So one of it is going to come in here. All right, come into here. The tube nucleus, of course, gone already lah. Okay, let's look at this diagram. So one of it, the male gamete is going to come in and it's going to fuse with your egg cell. So when it fuses, your male gamete is N, correct? Okay, this male gamete is N. Okay, male is this symbol. It's going to fuse with your egg cell. Your egg cell is also N, correct? In fact, N of it is N lah. Okay, so what are you going to get? N plus N. You're going to get what? Okay, what are you going to get? Can someone just maybe type in the comment? Okay, Muhammad Amal, thank you very much. Okay, I hope you enjoyed the lesson. Yeah, okay, so N plus N, you get what? You're going to get, okay, simple maths, right? Okay, 2N. 2N is already called a diploid. So you are going to get a diploid cell, all right? This is called a zygote as a result. So this zygote is diploid. Okay, so this is the first fertilization. So it's going to be 1 fertilization, all right? Don't forget, there's going to be another one. Okay, the second one. So there's going to be another male gamete, right? First, 1 male gamete already used. So you got the second one coming, all right, of course, together, all right? So the second one is also N, haploid, ma. Every gamete is haploid, okay? Now, this haploid gamete is going to go up there, okay? Now, the egg nucleus is already fertilized, so don't worry about that. But there's another 2 things that haven't been fertilized. In these 2 polar body, or polar nuclei, not polar body, yeah, polar nuclei. They are 2N, because N, there are 2, ma. So now your male gamete, this one, is going to fertilize that 2 polar nuclei. Okay, so N and another N. Okay, so you have, what is the total? What is the outcome after fertilization? At the fertilization, what are you going to get? Okay, let me see anyone. Yeah, okay. So yeah, sorry, correct. 2N, 2N. Now we're talking about the second one, right? Okay, now after the second type of fertilization, you have N, you get a 3N. Something which is not common, right? You don't get it in the animal kingdom. But in plant kingdom, we call it we call it polyploidy. That means more than 2 sets of chromosome is possible. You have triploid, you have a tetraploid, you have polyploidy, that is many sets of the chromosome. It's possible in the plant kingdom. It is not abnormal, not rarely abnormal. It's also possible. Yes, correct, 3N. So we call it triploid. So this is a triploid, and we don't call it a zygote. Okay, we're gonna call it endosperm. Okay, endosperm. Nothing to do with sperm, right? We just call it endosperm. This is going to be actually, what's the function of endosperm? It is going to be a tissue that is going to store food for the embryo. The embryo is from the zygote. That one is going to become your baby plant. Your zygote will become the baby plant. Okay, it's going to grow, become a baby plant. The endosperm is just like a storage of food. It's makanan simpanan. It's a storage of food for the embryo that is going to germinate. Even if it doesn't germinate, all right, it needs to be alive, isn't it? When it stays alive in the whatever, it needs food as well. So it's going to be providing nutrient, right, for the food for the embryo that germinates later. Okay, so that's why now you have 2 fertilization. So that's why I call it double fertilization for flowering plants. It's a concept that's never seen in the animal kingdom. It's not common in animal kingdom, but in the plant kingdom, it's quite common. Okay, we have tetraploidy. Okay, we have polyploidy, more than 2 sets, 3 sets, 4 sets even. Okay, so this explanation actually explains all that. Okay, you can go through this is a textbook, you have it, you can just have a look at that. So the 3 antipodal cells and the 2 synergid cells, they play no role in the fertilization at all. Nothing, all right? That one just like the polar bodies, it is just there, it doesn't do anything. So the most important thing, you remember the egg cell, which we're going to develop into zygote with after pollination, after fertilization, and your 2 polar nuclei, which is going to be the endosperm, is going to be triploid after fertilization. Okay, right. So now let's look at other things. All right, if there's no other questions, let me go to interesting things about formation of the fruit. That is very interesting. I find it. Okay. Okay, let's go to this one. What is the importance of double fertilization? Why do we have double fertilization for the flowering plants? Okay, first of all, double fertilization produces 2 things. One is the zygote and one is the endosperm. Okay, so first of all, the fusion of one of the male gametes with the egg cell, remember this is an N, this is an N, right? You'll get a zygote, which is how many N? Zygote is always 2N. Okay, this is going to be the baby lah. The baby, the plant that will grow when you plant your seed. This is the embryo. The zygote later on will form the embryo. And then the embryo, you have the plumule and the radicle. Remember, you learned that in Form 3, right? Or Form 1, I think, when you learn in your reproduction. So you have the plumule, you have the radicle, right? That is the embryo. The plumule becomes the shoot. The radicle becomes the root. That's going to form the body of your plant. Okay, then another usage, why we need this zygote here. Genetic information is passed down from one generation to the next, so that means ensures the survival of the genes or the species. Okay, it restores haploid condition in gametes with the formation of diploid zygote. Okay, in fact, I think this point better. Restores or maintains the diploid chromosomal number in the species of the plant. Because you have diploid, which is the gamete, add another diploid of the female, gamete, male and female. In the end, whatever you produce, your zygote will always be 2N. And this is normal because this is the somatic cell, the composition of the plant or the species. You must have diploid. Okay, so it maintains the diploid chromosomal number in the species of the plant. Okay, you must have N and N, then you get your diploid. Okay, so just like same thing like your animal kingdom, when your sperm is N, your ovum is N, you get 2N, and then it forms a normal baby with 2N. You cannot have 3N, 4N, and 5N, and so on. That would be abnormal for human. Okay, so here you get the diploid chromosomal number. 2N is diploid. Okay, another one is the formation of don't forget your endosperm. Okay, endosperm tissue is the 3N one. Okay, why you have 3N? One male, and then you have two polar bodies, two polar nuclei. I'll keep on saying polar body, yeah, polar nuclei. So N and N, you get 3N. Okay, what is the use of this? This tissue is used for the development of an embryo. So it's to provide nutrition, is to provide food, okay, for the embryo that is going to grow later on. In eudicots, that means plants which have dicot, that means they have the parallel vein and so on, okay, such as legumes. Legumes are those family of plants that have beans and nuts, kacang, pokok kacang, all right, all the kacang, kacang tanah, kacang whatever, okay, peas and beans and all they call legumes. Mangoes and mustard is sawi. We call it choy sum in Cantonese, choy sum. The endosperm is fully utilized by the embryo to develop before the seed matures. So this endosperm is already used up by the embryo before the seed mature. That means it's very quickly used up by this in this species, in these examples. Now, mostly in other examples like monocots such as coconut, wheat, barley, and corn, only part of the endosperm is utilized for development of the embryo. They only use up some of it, not the whole endosperm. Okay, the endosperm still remains in the seed, right? So the endosperm is not used up for the entire development of the embryo. Some of the endosperm are still there. They are stored in the cotyledon to be utilized during germination. So when it germinates, only they use the endosperm. If it doesn't germinate, that means the endosperm is still there. So that's why in the seed, in the grain, when we eat the, our beras, we eat the oat, we eat the barley, you still have the endosperm there. I'm going to show you a picture, okay, where's the endosperm. The endosperm helps the embryo to survive in the seed for a long time. If it's not germinating, the food is still there. So it's to feed the embryo. Okay, so that's why the endosperm is important. So that's why we need the endosperm. That's why you have the double fertilization. One is to make the embryo, or the zygote becomes embryo. The other one is actually to make the endosperm. Okay, now I want to show you some pictures that I found which is interesting. Okay, so let's look at the coconut. Where is the endosperm? Now you can see a picture of a coconut here. You have the whole fruit. Okay, when you see the word carp, C-A-R-P, carp is not the fish carp. This one carp is actually means the name of it's a Greek word, actually means fruit. Whenever you see the word mesocarp, pericarp, exocarp, sounds a bit like Thai. Thai sentence, everybody, every sentence they say carp, and all that, right? Okay, so endocarp. All right, now these are 3 layers. The whole thing, the fruit wall, is called pericarp. Okay, I'm going to introduce to you the formation of the fruit. Okay, later on I'll tell you also. Pericarp is the entire wall of the fruit. The ovary becomes the pericarp. Okay, but pericarp has got 3 layers. You have the exocarp. Exo means outside. The outside layer is tough. It's going to prevent entry of pathogens and virus and bacteria. So it's going to protect the whole fruit. There's the pea, the skin. Like you said, when we eat the mango, we cut off the slice of the skin, right? That is the exocarp. Okay, then the middle part, mesocarp. For the coconut, it's going to be quite fibrous. It's the sabut. We call it the sabut, right? Which we peel off. We don't eat if it's very, very old already. We peel off. But in the fruit like mango, the mesocarp is a fleshy one. That is the flesh that we eat, the orangey, yellowy color that is sweet. That is the mesocarp. That is the mesocarp we're eating for most of the fruits. That's the mesocarp. Okay, and then the inner part is called the endocarp. Endo means inner. Okay, that's going to surround the seed. So the 3 layers of the ovary wall actually is called the exo, meso, and endo. After that, you're going to get your seed. That seed inside that. For the coconut, the endosperm is the meat, the coconut flesh that we eat. So that is the flesh there. And then the liquid endosperm is the water, coconut water. Okay, so interesting enough for you to know, isn't it? So actually the wall of the fruit, it can differentiate into 3 layers, okay, after it's developed into a fruit. That's what happens after fertilization. Okay, so I've jumped the gun. Actually, I just went straight into the, talking about the fruit. Actually, I forgot to tell you, after the fertilization, your ovary will become the fruit, and the ovule, that means the seed, the ovule will become the seed. Okay, now let's look at other examples. Barley. Barley is yi-mai in Cantonese, yi-mai. Now you see where's endosperm? These are monocots. Monocot, like we say, endosperm, they do not get used up. So you see, endosperm is the food that is in the seed itself. This is what we're eating. We're eating the barley, the barley, the yi-mai is the whole biji there, right? That is the barley. And bran, you see the bran is the skin. The germ includes there, you have this what? You have that embryo inside there. Okay, that's the germ. So when you see all these different types of bread, wholemeal lah, okay, wheat germ lah, bran lah, so many types of bread, isn't it? Okay, I'm going to tell you what is difference there. So we look at the anatomy of a seed. Okay, I'm giving a lot of information now. This is actually very interesting. So let's look at jagung. Right, you see where is the embryo? Embryo is here, right? This is going to be the plumule and the radicle. This is going to grow into your baby corn later on, the plant. And the endosperm is the food inside there. It stores the food there. The pericarp is the outer wall of the ovary lah. Okay. Now then, this one. This is the what? This is wheat. Gandum, where we used to make what? Make flour. Make bread, make all this food that makes from flour and all that. Okay, so the seed is bran. The outer skin is bran. Endosperm is what we grind it into the powder, the flour. That is mostly endosperm. Okay, germ is including that your embryo inside there. All right. So bran, endosperm. Now we talk about bread. Let's talk about bread. Which is more nutritious, white bread or wholemeal bread? Can you tell me? Okay, I want some interaction with you. Which is more, of course, more expensive also and more nutritious, people say? What type of bread? Do you go for white bread or do you go for wholemeal bread or bran, wheat bran? Some even call wheat, don't know wheat germ or bran. Okay, which one? Which kind of bread do you go for? I'm not getting any, maybe I have to wait for a while. Okay, never mind. Now, refined grain, this is the one that we get for white bread. White bread we use from the endosperm only. The middle layer means we don't take out the bran, we take out the outer part of it, we take out the germ. Okay, I'm getting some response. Let me see. Yeah, wholemeal, correct. Wholemeal is more expensive, but of course it's more grainy, isn't it? Some people don't like it because not so smooth, very hard, a bit harder, a bit more texture, not so smooth, but it's actually more nutritious. Why is it more nutritious? Yes, whole wheat bread, correct. It because wholemeal, we call it whole grain because it includes the bran, the endosperm, and the germ. Everything included. So you get more vitamins and more minerals. Okay, rather than you buy white bread. If you buy white bread, it is only made up of the endosperm. The endosperm, the starchy carbohydrate middle layer. So they take away the germ, they take away the bran. I don't know how they asingkan, they have to separate it. Okay, so you get that white bread. That is the one. And it's not so nutritious because you're only getting one part of it. Many of these nutritions you get from is found in the bran, found in the germ. Okay, so it's better to take wholemeal bread. Okay, so this is the difference there. Always read the label, right? Always read the label when you buy things. You find the wholemeal actually includes the bran and includes the germ. You got more nutrition from there. Okay, but of course, children don't like it. My kid doesn't like it. They prefer white bread because it's softer. Okay, so that is interesting thing for you. I want to add in, right? If you just read the textbook, you don't get anything extra. So I try to make it more value-added lesson for you, so they learn more than just what the textbook do. Okay, so now for development now. All right, so we are running a bit late, but I think we just extend a little bit of time now. I just want to make sure that I cover everything. Okay, so now let's look at here. Are you still able to see me or hear me? Everything okay? Yeah, let me just check. Okay, my mic is still on. All right, okay, correct. Now, let's look at, I already accidentally added the comment there. Okay, to show Rachel's comment. Okay, and then let's go to this one. Development of seeds and fruit. Now, we already have double fertilization. From double fertilization, you have two things. Remember, you have your triploid endosperm, which is 3N. Okay, and you have the other one, which is, what was that? Zygote. The embryo is going to form the embryo. So now, let's look at what happens. The zygote, okay, now this is going to form the food lah, the endosperm lah. Nothing much to see that. It's going to form the mitosis is going to happen. After that, it's all mitosis. When you talk about development of the fruit or after fertilization, it's always mitosis already. Just the cell multiply, divide and divide. 2 become 4, 4 become 8, 8 become 16 and so on. So it's going to form, endosperm will just form the endosperm. Okay, the triplet endosperm will form the endosperm. And then for the zygote, what happens? The zygote will divide by mitosis as well. It forms two cells. One is the larger cell, one is a smaller cell. So why you have these two things? Okay, first one, the larger is called the basal cell, right? It's going to develop into a suspensor. Now, what's a suspensor? Suspensor is a part of the tissue that's going to hold and stick it to the wall of the embryo sac, the wall of the seed lah. Okay, so this one is the basal cell, the bigger one, and the smaller one is the terminal cell. All right, so now the bigger one, I find the label here slightly a bit confusing, so I'm going to cancel this basal cell. The suspensor is actually referring to this whole part here at the bottom here. Okay, so it's this one. This is the suspensor. You see the elongated part here. This is the suspensor. This is the suspensor, okay? While the top part here is going to form the embryo. The upper part here, okay, I'm going to color green. This one is going to form the embryo, okay? So the bigger one, the bigger cell here is the suspensor. This one will become, the bigger one is going to form the suspensor. It's going to anchor lah. You see, later it's going to develop this piece of thing here. No, I'm going to put green. It's going to confuse you. I'll put red lah, this one. This one is going to stick it there. See, stick it to the wall of the seed there, right? If not, it's going to come off, right? It's just stuck to the wall of the seed. We're looking at the seed. We're not looking at the fruit. It's the seed. Still the seed. Okay. So now, the other part is the smaller one, right? The smaller cell there. The smaller cell will become the embryo. That will have three parts. The plumule, remember, the plumule will turn into the shoot, the radicle will turn into the root, and then the cotyledon will store food. So this three part is the embryo. Okay, and the endosperm is the outside there, that all this part here, right? That is going to store food as well. So you have the endosperm there, you have the embryo there. So two things there. Okay, the embryo will be part of the germ. The endosperm is the part, majority part of the thing that we eat, the grain. Okay, so now let's talk about this whole thing is going to be an ovule. It's going to become the seed. Okay, now the seed here, there are many types of, and the ovary is going to become the fruit. Okay, ovary will develop into a fruit. And what happens to the stigma and the style? So if you look at the fruit, this is a flower, an apple flower. The apple flower, once it already fertilized, where all the pollen grain has come in and then everything, fertilize the male and female, you get the endosperm, everything. This part is going to disintegrate. The stigma is not needed anymore. It will dry up. It will die. It will dry up. And of course, the stamen all not needed. The petals will drop off. Everything will drop off. The sepal will drop off and everything. You are just left with the ovary wall. All right, it will become the fruit. And the seed there will become the seed. And for the case of apple, in fact, this is not a very good example. An apple is a false fruit. Why do we call it a false fruit? Very interesting. Not all fruits develop from ovary. Okay, we always have the concept the ovary is the wall of the fruit cell. Actually, the ovary, the fruit that we eat, sometimes do not come from the ovary. Sometimes it comes from the receptacle. Like for the case of apple, it's called a false fruit. False fruit because it's not really coming from the ovary. It actually develops from here. This is the receptacle. This part that expands, the part that we eat, this one, the fleshy part, actually is the receptacle that holds the flower. The flower has dropped off. Okay, and this is the ovary, the seed that's inside there. So it just envelops the what do you call it, what do you call that part here? Just I mentioned the receptacle here, it envelops the whole part of the bottom of the flower, and it just bloats up and you get an apple. Okay, that's interesting. Okay, so I want to show you a true fruit or false fruit. Okay, so now first of all, here again, I'll just remind you, ovule will form the seed. This is for development of after fertilization. The ovary will form the fruit. The ovule will form the seed. The integument are the layers that cover the ovule in the beginning, right? It's going to form the seed coat, testa. Okay, like the seed, the skin of the seed, we call it testa. Okay, now let's go to interesting part about fruit. There are two things actually, whether a fruit is true or false. Okay, I have so many things to show you. In fact, I'm going to show you. This is just now the suspenser. Okay, the development. Sorry, I retract a little bit back. Okay, so is tomato a fruit or vegetable? I already mentioned to you earlier. Ask yourself, does it have seed? If it's a seed, that means it must have fruit. It must have a fruit, then it must have flower. So it's definitely a fruit. Okay, a lot of people argue, is it seed or a vegetable? They're not too sure. Okay, now this one, remember the basal cell and the terminal cell? So this basal cell will form the suspenser. There you see. And this terminal cell will form the embryo. So not to be confused, this is a suspenser which is going to stick to the base of that, the ovule inside the seed there. Okay, now let's go on quickly. What is the difference between a fruit, true and false? Now let's go to see a true fruit first. What is a true fruit? A true fruit can be defined as a fruit which is formed from the fertilized ovary. Definitely come from ovary one. That's called a true fruit. Okay, a true fruit definitely developed from a ovary, just like mango. Okay, Puantang, hi, good afternoon. Okay, so mango comes from a true the flower, the ovary. Once it develops, you get a mango. Now, any true fruit will always show the following: the pericarp. Remember the pericarp, the three layers. You have the exo, meso, and endo. Pericarp is the ovary wall. Okay, and usually the part that we eat, the fleshy and the soft and the sweet part, is the mesocarp. Okay, and then you have the seeds, they're fertilized. They are ripe. They are the fertilized ovule. Means the seed are actually from the ovule, and then the fruit is actually from the ovary. Okay, and then the remains of the star already dropped off. Stigma already dropped off. And then you can see the scar. That is a real fruit. So what are the real fruits? Mango law. Okay. Orange law, papaya law. Many of the fruits that we eat actually are true fruits. Now, what is a false fruit? Interesting enough. A false fruit can be defined as a fruit which is formed from the ripened ovary along with some other parts of the flower, like the base of the receptacle. Like I said, the apple. It is not from the ovary, but the receptacle just kembang. It just expanded, and then it can cover and envelop, and then that part becomes soft and fleshy and juicy. That is not from the ovary. So apples, banana, cashew, the yu tao, the cashew there, that one nice to eat one, expensive one. Okay, strawberry, they are all examples of false fruit. There are some of the flesh, it's a fruit in which some of the flesh is not derived from the ovary, but some adjacent tissue. There are other tissues that are adjacent to the ovary, okay? So they are called it false fruit. We call it pseudocarp. Pseudo means false. Carp means, again, I said fruit, okay? So examples, strawberry, pineapple, mulberry, apples, pears, and so on. Okay, so they are false fruits. So here, again, another similar explanation. They are formed from other parts. So let me tell you where are the other parts. So they can be formed from the base or the receptacle, example, the apple, the perianth. Perianth is the outer part of the flower. So here, it can be the sepal. Sepal is the part that covers the, the flower before it blooms, the sepal, okay? The perianth, or the petal part. The whole thing is called corolla. The whole, 5 petals there is corolla. And then one of it is called petal. So the whole thing is called corolla. Sepal is one piece. The whole thing is called calyx. So that's called the perianth. Okay, so it can be developed from there. Thalamus is which part? Okay, actually, no need to go into all these details. It's not really needed. But as long as you know it does not develop from the actual ovary, it's good enough. Okay, too many things to give you actually. I don't want to overwhelm you with too many facts. Okay, just to let you know that enough for you to make it is interesting enough. There's so many difference and variation. Okay, so here examples are cashew nut, developed from peduncle. Peduncle is the part that holds the flower. Okay, apple, pear, gourd, cucumber also falls. Develops from the thalamus. Jackfruit, nangka. Jackfruit is called a nangka. And pineapple develops from the entire inflorescence. Inflorescence is a group of flowers. Okay, a group, not only one flower, many flowers. Then after that, they all merge together to get one whole big thing called the pineapple. Okay, so these are the examples. Jambu air also, the 水庸. Okay, jambu, I call it water apple. In English, the jambu is called water apple. The Syngium jambos is the scientific name lah. Figs and all that. There's so many different. Okay, let me check. You got some response there. Coconut. Coconut is, let me see. Coconut comes from the flower. Coconut is a real fruit, I think. Let me check. It's a simple fruit. Coconut is a real fruit. Yeah, wait. Yeah, I have it here. Now, types of fruit. Let's go to the last part. Interesting enough. You have different formation. Fruits generally, of course, come out of fertilization. But like I said, it develops from different parts. Not always from ovary. So that's why you have so many different types of fruits. Different categories of fruit. We have 4: simple, aggregate, multiple, accessory fruit, or we call it the false fruit. Okay, let's look at the 4 types. Simple, just I managed to show you the false one. Okay, let's look at the simple fruit. This diagram helps you to understand. When you have a simple fruit, it comes from one flower. You can see it's only one flower, right? One flower with a petal and so on. And there's only one pistil or one couple. Okay, one flower, one couple. It is a simple fruit. So you have example pea, the chang tao, the pea, the green peas. Okay, not the mung bean, the green pea, the one in the pot one. This one, this is the green pea. This one, the pea. I'm talking about this pea. Okay, not the lok tao. Okay, orange, tomato, peach, coconut. Yeah, coconut is a simple because one flower, one couple. Okay, so okay, next, aggregate. Aggregate is one flower. You see, there's only one flower, but it has more than one couple. So more than one couple. Sometimes one flower can have more than one couple. Okay, in fact, I call it pistil. If they join together, it's called pistil. So you have examples: raspberry, blackberry, strawberry. Okay, I'll show you an example. Why they call it that? You see here? This is your raspberry. You see, you eat the fruit, you eat this whole thing, right? You think it's only one fruit. Why actually? Actually, it is little flowers. Different flowers that have actually joined together. So you can see this one little thing. One little thing here with a little thing, the head sticking out. That was from the stigma. Okay, that didn't totally drop off lah. So this is actually one from one flower. So you have so many flowers grouped together, right? Actually, sorry, one flower, but many different couple. Okay, so one flower is come from one flower, but different couple. So that's why one flower, many couples, you get a raspberry, blackberry, strawberry. Okay, I want to show you a strawberry. Everybody gets an idea. Strawberry, you see the seeds outside there, right? Those are not seeds. Strawberry seeds are not seeds. Okay, what do you mean by that? Can anyone guess what, if I say they're not seeds, what are they actually? I just want to check. Can you give me a try and answer this? Why are they not seeds? The things that you see here, this one, there, this thing, right? All these little things on the surface of your strawberry fruit. I cannot see here because it's red color. Let me change the color to a different one. Okay, blue. Right. See all this? All this biji thing, right? Why are they not seeds? Can anyone tell me? Rachel, Quan Tang, maybe you can try. Sorry. Yeah, why are they not seeds? All these little things that you'll be eating, you're thinking you're eating the strawberry seeds. Coconut is a true fruit, if I'm not mistaken. They are, yes, Xiaoyi, you're right. Each one of them is a fruit. So small, the fruit. And the one that developed, the whole red thing that developed is not the ovary. So that little thing that you see, okay, this is actually an ovary. And if I say it's an ovary, that means inside must have seed also. The seed is even smaller. Okay, the seed is even smaller. So actually, a lot of things we find that we always, we see it, we always think the way we, but actually they are not what it seem to be, right? Interesting enough. That's why I think today's is very interesting. Yeah, it developed from somewhere else. So it is the receptacle. Yes, possible. Yeah, I've got to check it out on that. So it is not what do you call, it's not from one flower, okay? It are not from one couple. One flower, many couples, right? All these aggregate. Now, multiple is like how? Many flowers that flower has got, that plants many flowers, but they all fuse together. Many flower means many couple lah. Many flower, many couple, and they fuse together to form a big thing called the whole fruit, which is the example pineapple. So each one that you see, pineapple has a lot of eyes, isn't it? Each eye that you see is actually, this I put red, this I put red color. Okay, this one, each eye that you see is actually from one flower. Yeah, each eye. That's why you see, the pineapple very difficult to eat because of the eye. You have to cut off the eye, isn't it? So each one of it is actually from one flower. And of course, there is one pistil, one couple inside there. And they all fuse together to form the whole mass there called the pineapple fruit. That's called a multiple fruit. Okay, so there's slight difference there. So you want to remember, you remember this one. This is important so that you know the difference there. It's whether we got how many flower, how many couple. One flower, one couple is simple. One flower, many couple is aggregate. Many flowers, many couple is multiple. And accessory fruit is the fruit that's developed from tissues other than ovary. So apple, strawberry is also under accessory fruit, false fruit. Okay, cashew, another one is cashew. Okay, there are many figs. Okay, fig. So you see, it's so interesting. Right, for the pineapple, for the apple, the remains, this is you always the top, but this is actually the top, the remains, the stamen, the bottom is actually the top because the fruit is like that. The fruit actually, you always think it's the top, this is in fact the peduncle, the part that was, so the whole fruit actually is the balik kan. The bottom part is actually where there was the stamen and the styles that already dropped off. Just like I said, the orange. Right, I see the orange, I have got this thing, isn't it? I have got this part here. And the bottom here, normally you have one little lubang there, right? When you have the, navel oranges. This is actually where the flower, the stamen was, last time. The ovary, the style and so on was here. And then this is actually the tangkai. So you see, the whole fruit, actually was the balik. Like that. Interesting enough, we always get the wrong impression. Okay, so any questions here so far? I think it's interesting enough. Yeah, receptacle, yeah, that was right. So just want you to know that so many different types of variations because of the things, are grouped together, how flowers are formed, different flower, different couples and so on, couples. Okay, now let's go to the last part. Importance of seed survival. Again, there's not much there, so that's why I didn't want to put it into the title there. So why do we need seeds? Okay, of course, seed is a way to propagate to make sure the next generation lives on. This carries the genetic information from the parent. Okay, so it's to maintain the survival of the plant species. If you don't have seeds, all right, you have to rely on vegetative reproduction. For plants, it's no problem. You don't have seeds, you still can have the next generation because they can multiply. They can actually reproduce vegetatively very well. Okay, no problem with that. But of course, animals is slightly different, very difficult. So far, vegetative reproduction is not really viable for animals. Okay, now seeds contain embryo that germinate to form seeds. So you have to have the seed which contain the embryo, and it is the embryo that's going to make the baby plant. If there's no embryo, there's not going to be a baby plant. All you have is the endosperm in it. That means, if you imagine there's no zygote, okay, there's only endosperm. You only get the food only. You're eating the seed for the food. There is no embryo. It's not possible to germinate from that. If a seed does not have embryo, that means it's not going to be a baby plant. Embryo is just like a baby, okay, waiting to grow. Inside the seed, the endosperm, the tissue or the content is the source of nutrient. So the endosperm is a source of nutrient to supply energy to the embryo that is going to germinate. Okay, and another 2 more points is more for protection. So seeds are enclosed by testa. Testa is the kulit biji. We call it testa. In scientific terms, it's called testa. It's formed from the integument. Integument of the layers of the tissue surrounding the embryo sac. Okay, it's strong, it's water impermeable to prevent the seeds from spoiling, to keep out the pathogens, okay, the attack of virus and pathogens, the bacteria and so on. Okay, it also enables the seed to be stored for a long time. So seeds usually don't expire. And the good thing about it, you can keep it in the fridge. All right, you can put it in a dry place. The seed inside, it's frozen in time. Okay, the seed is frozen in time. Once you put it in the suitable conditions where you have, remember, you have 3 things. You need to have water, you need to have oxygen, okay, as air or specifically oxygen. You need to have the correct temperature. These are the 3 things that you need for germination, not sunshine, not light, because the light is only needed when the seeds come, when the leaves come out. That is only about maybe a few days later, okay? Otherwise, if you just germinate, it's just good enough to have water. You need to have the correct temperature and oxygen. Okay, so it is frozen in time. Just to say a very simple way is your seeds can grow anytime. You give them the correct conditions, they will grow perfectly. Okay, and lastly, all right, seeds have special features that they are light, so they can be dispersed, they can be brought by the wind, they have spongy tissue, they do not spoil, so they are easily dispersed to further places so that they do not get they do not accumulate in the same place where there's going to be an intense competition. So the fact that they can be dispersed by animals that eat the fruit, the seed will be taken away, and then they, of course, sometimes they eat into the stomach, right? And then when the animal goes to another place, and then they excrete, that means they shit, right? They shit, and then the seed comes out. That gives them another chance to be grown in a different place, further away from the competition. So it's good to have seed, and they do not get what? They do not get digested. So you can eat a seed, a bird eats the seed, right? The seed, the testa is very strong. They do not get damaged by the enzyme of the stomach of the animals. So when it comes out, it comes intact. That is one important thing about the seed. It ensures survival so that they have a good chance of germinating in a further other places, so that they give a higher chance for the survival of the species. Okay, so that is my lesson for today. I took a little bit longer, but I think it was worth it because I have a lot of information to impart to you and to give you a better idea of the lesson. Okay, so if you want any of these diagrams, you can actually just pause the video and you can actually, copy and paste and so on. You can take a picture and all that. You can have copy the notes and so on. So no problem. All right. Okay. So any questions so far? I think I don't see any. Okay. So if there's no further question, I would like to end the lesson now. Okay. Hopefully I can have another lesson soon. All right. We're going to have online classes starting from, very soon. And I'm going to give you more lessons, hopefully. Okay. All right. So I will say goodbye now. We'll end off now and hope to see you another time. So don't forget to do your homework. Okay. Right. Thank you, girls. Welcome. And I hope to see you another time. For those who are new, who have not subscribed, okay, if you do subscribe, the advantage is you're not helping me, or of course you are helping me, but it doesn't matter. Is you're actually, whenever I have a class, if you don't get notified, if you subscribe to me and you click the bell button, you will get a message that I'm having a class. So you will always be notified that the class is on. Okay. All right. So I'll see you next lesson. All right. Okay. So bye-bye. See you soon. Bye-bye.